Good morning. It's a great pleasure to be here and to start off this session this morning talking about targeted inhibition of signal molecules as novel therapy for CLL. Just some acknowledgements at the outset. I do want to recognize the Mayo team that did a lot of work on Axl receptor tyrosine kinase that I'll describe to you. In a moment, in particular, Ashish Ghosh, also Tolera Pharmaceuticals for providing their drugs for us to test, and the support from the NCI for both Dr. Ghosh and myself. Learning objectives this morning are really threefold. First, to identify for you the signal molecules that we now know are critical for both normal and leukemic B-cell survivalship. I'm going to review the necessary preclinical data that supports the potential efficacy of signal molecules in CLL therapy, and this is really kind of a primer for you in terms of how we would proceed to want to test certain drugs that target in CLL. And at the end, we'll do a brief discussion on how we might look at combining these inhibitors of signal target molecules as strategies for future therapy. Now, signal molecules as targets have always been attractive, and that's because protein kinases and lipid kinases are very good choices as targets of signal transduction inhibition, and that's because these pathways are often related to the pathogenesis of lymphoid malignancies. And in particular, we're aware of the BTK and PI3 kinase with their prototypic drugs Ibrutinib and Idelalisib, and you're going to be hearing a lot about this in other talks this morning. I'm really going to focus on the basic biology of these two targets, and then I'm going to talk to you about a unique receptor tyrosine kinase that we have discovered is present on CLL B cells, Axl, and it is also targetable. Now, signaling pathways can look very complicated, and unless you try to pay attention to the core components, sometimes it can appear rather daunting. But let's turn to the B-cell receptor, and just suffice it to say that really all you need to remember is that the antigen, once it binds to the BCR, activates Lyn and Syk, which in turn activate NCK and BCAP, which are linked to the CD19 molecule. When that happens, BTK and PI3 kinase delta are activated, and for BTK, there's a series of effector molecules that are activated, subsequently resulting in NF-kappa B and MAP kinase being activated. And for PI3 kinase, PIP2, once it's activated, PIP2 is converted to PIP3, and the master signal molecule, AKT, is also turned on with subsequent downstream effectors that turn on four other pathways, in particular P53 and mTOR. The important point is that all of this activation ultimately results in enhancement of survivorship, proliferation, chemokine synthesis, and metabolism that's critical for leukemic B-cell growth and survivorship. And many of these pathways are, of course, shared with the normal B-cell. Now, just to underscore the importance of the B-cell receptor, it's important then to remember it's present on all B cells, of course, has two main functions. It recognizes foreign antigen, and it initiates the signaling pathways that result in selection of B-cell maturation, survivorship, proliferation, and participation in the immune response. Now, as I just told you, BCR signaling is mediated by a number of proteins, and that, of course, includes PI3 kinase delta, BTK, and other signal molecules, but we're really going to focus on the bolded ones, PI3 kinase delta and BTK. Now, what about BTK? This is a TEC family non-receptor tyrosine kinase that plays a dominant role in B lineage lymphoid cells. It's really a key molecule critical to normal B-cell development. Well, how do we know that? Well, there's really a brilliant observation in 1952 by Dr. Bruton, who described the inherited syndrome X-linked A-gamma, and it turns out that XLA is a human immunodeficiency syndrome where there is a failure of pre-B cells in the bone marrow to develop into circulating mature B cells. And BTK has this association with the pathogenesis of XLA supported the critical role as a key regulator of B-cell development. And this discovery now almost over 70 years ago, some 70 years ago, is now playing a role in our treatment of CLL patients. So We're all aware that various drugs were developed to inhibit BTK. In particular, we're focusing on Ibrutinib, formerly PCI 32765. This is a potent, irreversible, covalent inhibitor of BTK. You see the chemical structure here. The Ibrutinib is able to bind to the TK region, and it irreversibly binds to Cis481. And this has an extremely low nanomolar concentration, so high selectivity for BTK, although not complete. Herman showed some time ago that BTK protein levels in CLL B cells exist, somewhat variable but definitely detectable BTK levels. Subsequently, it was shown by Honigsberg that Ibrutinib can downregulate BTK, but notice it does not downregulate the BTK total protein, it just downregulates, here in increasing concentrations of Ibrutinib, the phosphorylation status of the BTK. Work also by Herman showed several years ago that Ibrutinib in vitro does induce cytotoxicity, and quite frankly, it's modest, but it is concentration and time dependent, increasing over 72 hours, and I ask you to remember this for some other components of my talk later. Importantly, they also showed that this induction of cytotoxicity was independent of risk status, that is IVGH mutational, and also high or low risk fish. Now, there's a lot of other preclinical data I do not have time to get into in detail, but there's been a tidal wave of publications about how Ibrutinib can disrupt the environment. Remember that the CLL B cell loves to nurture itself through interaction with the microenvironment. I'm just highlighting some of the aspects. It antagonizes BCR and CD40 ligand-mediated signaling and proliferation, abrogates protection mediated by the microenvironmental factors, reduces CLL chemotaxis towards chemokines, nurse-like secretion of chemokines is reduced, and disrupts adhesion of CLL cells to stroma, to various substrates in the stroma. And there's also very important data for Ibrutinib in mouse models. Kill et al. Showed a few years ago in a CLL mouse model that with knockout of the BTK, there was no leukemia. Woyach and co-workers have shown either through genetic inactivation of BTK or the use of Ibrutinib, and here's a figure from that paper, here with the mice exposed in a TCL1 mouse model, a very aggressive mouse model, that leukemic survivorship is significantly enhanced, that the mouse survivorship is enhanced. Now, there are some off-target effects for Ibrutinib. It does inhibit interleukin-2 kinase, which is an essential enzyme in Th2 T cells. The potential benefit of this is that it can shift the balance between Th1 and Th2 and allow for a dramatic anti-tumor response. And recently, Soon et al. Have reported that there's improvements in humoral immune status, particularly IgM and IgA, for Ibrutinib-exposed patients. There are some off-target effects. Unfortunately, there are defects in platelet function related to platelet signaling defects. There is an incidence of atrial fibrillation, probably related to inhibition of cardiac PI3 kinase AKT signaling. And there may be a decreased efficacy of anti-CD20 antibodies as mediated by natural killer cells. Woyach has also shown a very beautiful paper in the New England Journal that long-term exposure to Ibrutinib, even with responses in CLL patients, can induce mutations that result in drug resistance. And there were 4 patients in this report that had a cysteine to serine mutation, which results now in reversible inhibition by Ibrutinib of the BTK. And the PLC gamma mutations appear to result in a hypermorphic, that is an excessive functioning of PLC gamma 2, thus resulting in BTK independence for CLL B cells. So in summary of the BTK, we know that inhibition targets a key signaling molecule vital to its survivorship, extensive in vitro and preclinical model efficacy, and we all know that clinical trials have shown dramatic results. You're going to hear more about that. The caveats are at least drug resistance and off-target effects that I've listed for you. Now, what about PI3 kinase delta inhibition? Well, it turns out that PI3 kinase dependent signaling pathways can be activated in a variety of ways. Through activation of the BCR, CD40 ligand, chemokines, or adhesion receptors, PI3 kinase delta is activated, and AKT at least is activated, resulting in enhancement of these now familiar functional pathways that are clearly related to leukemic B-cell progression. Now, there are a number of PI3 kinase inhibitors in development, the isoform specific, the PI3 kinase delta, in particular the prototypic drug Idelalisib. There are pan-PI3 kinase inhibitors, and now dual inhibitors targeting both PI3 kinase and mTOR. PI3 kinase delta is the isoform primarily involved in BCR signaling. And this is important to remember, is present in circulating lymphocytes, neutrophils, and monocytes, and in lymphoid tissue throughout the body. But Idelalisib has been shown, by the way, this was previously Cal 101, that it has high selectivity for the delta isoform. And how do we know that? Brian Lanuti published several years ago using an in vitro protein kinase-based assay where he looked at inhibition of these isoforms. You can see the IC50 is some 110-fold more potent in inhibiting PI3 kinase delta than the other three isoforms. Now, can this then result in targeting of the PI3 kinase delta in malignant B cells? Brian, in the same paper, showed that constitutive AKT activation, shown in the inset for both CLL and mantle cell, can be inhibited by exposure to Idelalisib, and then subsequently with stimulation of CLL or mantle cell cells using CD40 ligand or BCR cross-linking, shown in the insert, again, Idelalisib significantly results in diminution of AKT activation. PI3 kinase delta inhibition by Idelalisib can inhibit chemokines CCL3 and 4. When you culture nurse-like cells with CLL B cells, you see enhancement of CCL3 and 4 in the green column, but note that with increasing concentrations of Idela, there's significant reduction to these chemokines. You can also reduce CLL chemotaxis and migration. Here in the same paper quoted here, you see that chemotaxis to CXCL12 or 13 is significantly reduced by Idela. There are off-target effects. This is a recurring theme in my talk. There's both bad and good. The bad is, of course, the increased incidence of diarrhea and colitis, and by the way, this has been seen in PI3 kinase delta knockout mice. Transaminitis, pneumonitis can be seen. There are some good things that occur with these off-target effects. Targeting PI3 kinase delta does not inhibit ADCC mediated by anti-CD20 antibodies, and some recent very elegant work by Ali et al. In Nature has shown that there may actually also be a stimulation of anti-tumor responses with Idela inhibition, with PI3 kinase delta inhibition, where there is a reduction of T regulatory function and an explosion of cytotoxic T cells. So in summary of the PI3 kinase delta with Idela as the prototypic drug, we know it inhibits key signaling pathways in CLL B cells and mantle cell. There's a decreased activation of at least AKT. This is associated with key reductions in pathways that the CLL B cell needs to survive. And it does provide, I think, for preclinical rationale for use in treatment in clinical trials. And now, of course, it's approved for use. Let's end up finally then with our own studies on Axl RTK and how we think it may be a promising target for CLL therapy. Now, why would we look at other targets given what we have now? There are issues. The current BCR signaling pathway inhibitors obviously are promising, but they're not curative alone and resistance develops. There are issues for patients who discontinue ibrutinib that I did not have time to get into. There are low-grade but chronic toxicities, and of course there have been reports of the emergence of Richter's syndrome. So our strategy for searching for other targets was to see whether CLL B cells express other constitutively active RTKs and their functional implication in survivorship. If present, we also wanted to know if they shared the same common signal mediators that are listed here that BCR utilizes. And in fact, we have detected constitutively active Axl RTK and also basic fibroblast growth factor receptor 3. I don't have time to talk about the latter, but I will talk about Axl RTK. So this is just a flow to show you that there isn't much Axl RTK on T cells, if at all, a little bit on normal Bs, but we see fairly robust expression of Axl RTK on CLL B cells, but not on their T cells. When we do immunoblots, you can see phosphorylated Axl, variable amounts, but detectable on CLL B cells. You probably can't see this very well, but when we do confocal, these Axl RTK molecules are clearly in the membrane of the CLL B cell. It turns out that Axl RTK is a member of the TAM family, tyro3, Axl, and MER, and this is a specific family of RTKs. The word Axl was derived from analexito. I apologize for my Greek, but it's Greek for uncontrolled. And the human Axl gene is located in chromosome 19 and encodes a protein somewhere between 100 and 140 kilodaltons, and its ligand is Gaz6. It turns out there's been a fair amount of work of Axl in other human cancers, and depending on the cell type, Axl initiates various signaling pathways that we now all have heard about in my talk that clearly could be relating to the leukemic B cell. And this is mediated with a spectrum of signaling molecules, but note that these molecules are common to those that I mentioned to you on the BCR. And when Axl is overexpressed, at least in solid tumors, and actually even in AML, it is associated with very poor survivorship. Now, we went on to look then at the association of Axl with other cellular kinases and lipases. The strategy for this was to make lysates and then immunoblot, and in fact, we found SRC, SYK, PCL-gamma2, ZAP, and LIN in these lysates. We then immunoprecipitated the Axl to see if these molecules co-localized with the Axl itself. And this is data that we have now published showing that in the IP, you can see the Axl, but you can also see expression of PI3 kinase, SRC, SYK, PCL-gamma2, ZAP70, and LIN. And what we think currently is that Axl does provide a docking site for these key signaling molecules. Now, Axl inhibition, we have found, and again published recently, induces robust apoptosis in B cells. Now, a drug that we obtained from Tolero, TP093, you can see downregulates the phosphorylation of Axl in 4 different patients. But what's more important here is look at the kill curve on CLL B cells. It's at nanomolar doses, if you can't quite see that, maybe you can on your iPad. And this is in low-risk fish. When we look at high-risk fish, which is, if you will agree with me on this, 17P and 11Q, even here the kill curves are at nanomolar doses. And this is over 24 hours, not a prolonged exposure. We've also looked at apoptotic proteins and pro-apoptotic BIM, and what we find is with TP0903 exposure, we see a reduction in BCL2, but predominantly in XIAP and MCL1, and those 2 anti-apoptotic proteins are key to leukemic B-cell survivorship. And we've also shown that there's an increase in BIM with TP0903. So here you see a downregulation of its transcriptional regulator, FOXO3A, excuse me, and a upregulation of BIM. So what you have is a combination, a seesaw, if you will, of reductions in apoptotic protection and increase in pro-apoptotic molecule BIM. So in summary of this work, the CLL B cells express constitutively active but variable levels of Axl. Axl acts as a docking site for multiple non-receptor RTKs that I've listed here for you again. Inhibition of Axl induces robust apoptosis even in high risk. And as I showed you just now, Axl inhibition does result in key apoptotic proteins that modulate survivorship. Now, just to end up briefly with some work that we've done in looking at synergy with other signal inhibitors. There's been a recent report by Martin that signal inhibitors can synergize. For example, Idelalisib and Ibrutinib can synergize to control BCR-controlled adhesion. So what we did is we took a TP0903, the Axl inhibitor, and combined it with an irreversible Ibrutinib BTK inhibitor and a reversible one that we obtained from Tolero. And what you see here are the kill curves where I'll just point out, here's the killing in the red line for TP0903, some upregulation with Ibrutinib, but a very much more dramatic upregulation with the reversible BTK inhibitor. And we've done what's called a combination index analysis, which looks at, again, at synergy or additive impact. And in 6 out of 10 patients, we do see complementary activity with the combination of the TP0903 and the reversible inhibitor. So I think this is my second last slide. Therapeutic opportunities that we see here. Axl inhibition can induce robust apoptosis in vitro. RTK inhibitor in combination with a reversible BTK inhibitor has even more impact on the majority of patients, either additive or synergistic. And these are all orally bioavailable and would be candidates for clinical trial testing. This is my last slide, and basically summarizes what I hope I've convinced you of. Signal molecule transduction biology does have plenty of rationale preclinically for targeting. We have to figure out some other targets. There is no perfect target yet. There are also many issues, and again, I'm only touching the surface here, that include off-target issues, drug resistance, and some other issues that I would relate to, I just did not have time to talk about, are how to best combine these inhibitors, further enhance the selectivity of the target, develop inhibitors that minimize drug resistance, and of course, to prevent emergence of more aggressive CLL or Richter's transformation. I'll stop there and thank you for your attention.